Professor Sajid Maksud from the College of Agriculture and Veterinary Medicine, and Dr. Mohammad Tawkir Alam from the Department of Biology. I would like to thank you all for your time and all your input, which has definitely added value to or improved the output that Priya has managed to produce. Now let me briefly mention the procedure to you. First, Priya is going to give us a 30 to 40 minute presentation. She's going to keep us occupied for 30 to 40 minutes and hopefully be very exciting. And followed by that, the audience will get an opportunity to ask a few questions. And once you have done with the questions, we will politely ask you to leave the room so that the examining committee can ask a few questions to Priya. Now once that's done, and the committee will ask the candidate to leave, and we will deliberate and come up with a decision. And once the decision has been made, we will invite everyone back in to announce the final verdict. Now that's about the procedure. Now finally, let me talk briefly about the person, the candidate itself. Now Priya is one of the most devoted, hardworking person I've seen for a very long time. She has been in the department for over 10 years, much longer than many of us over here, and one of the most long-serving research members of the department. Now, over these 10 years, Priya has managed to produce over close to 25 publications, most of those in Q1 or top-ranked journals. And just in the last 6 years she was doing a PhD, she has managed to produce 15 publications, which is much more than what I managed to do during my PhD days. So that just shows the amount of effort that she has put into the work and into her PhD. Now, this presentation and the defense itself, the thesis itself, will not do justice to what she has done. Like I said, she produced 15 publications, and the work she's going to present is just about 3 of those publications. So we look forward to hearing from Priya. She's going to keep us occupied for the next 30 to 40 minutes. Priya, the floor is all yours. So, Managing, are you able to hear that? You hear clearly? All right.
So, good morning, everyone. Thank you, Professor Ranjit, for your kind introduction, and thank you, everyone, for joining me here. Today, I am honored to present my PhD research on the topic therapeutic role of bioactive peptides derived from hemoglobin. And this work explores how naturally occurring peptides in our body can exert therapeutic benefits. And going to the outline of my presentation, I will start with an introduction, followed by the hypothesis, research objective, materials and methods. And this research I have done 3 different studies and key findings from 3 different studies, summary of my results, and finally I would like to address the future studies and limitations of my work. So let us begin with the basics. What exactly the proteins and peptides are? So everyone will be familiar with the term protein, right? The protein are biological macromolecules that are made up of longer chain of amino acids, and they are central to all biological processes in our body. And coming to the peptides are literally shorter chains of proteins which is composed of fewer amino acids, often fewer than 100 amino acids. So even though they are shorter in size, they have significant impact in the biological system of our body. So let us explore what peptides actually do and what is the therapeutic purpose of these peptides. And talking about significance of peptides, just as proteins, peptides can also act as hormones, neurotransmitters and signaling molecules. And one of the most interesting facts is that they can represent a discrete family of pharmacological substances which have advantages over the small molecule drugs and biologics such as antibodies. For example, they are having lower toxicity, higher affinity and specificity, and they can specifically block the protein-protein interaction due to their larger size and flexible backbone for small molecule drug fail to do. And they are having lower immunogenicity and reduced production cost compared to the biologics or antibodies. And I would like to take you through the peptide therapeutic history. So the usage of peptides as drugs doesn't begin recently. It has a history of more than 100 years. Back in 1922, the first peptide drug insulin was introduced. And later in 1950s, 60s and 80s, so many advancements were made which helped to accelerate the discovery of peptides as drugs. And coming to the 21st century, there are more than 100 peptide drugs in the market which for various metabolic disorders such as diabetes, cancer, infection, and even for the COVID-19. And talking about the peptide drug market, based on the Peptide Therapeutics Global Market Report, in 2025 it is about $49 billion US, which is expected to raise to $76 billion in 2029. And looking into the peptide drugs approved by FDA over years, starting from 1940, we can see that there is a strong upward trend in the approvals of the peptide drugs. And these all data highlights the growing importance of peptides in the field of modern medicine, and they are coming to the mainstay of the modern medicine. So given this importance of the peptides, let us see from where these peptides are mostly obtained from. So they are obtained from bioactive peptides. So what exactly the bioactive peptides are? They are the short peptide sequence which consist of 2 to 50 amino acid in length, and they will be embedded in the parent protein. And one of the important interesting factors that biological activity of these peptides may or may not resemble the parent protein. For example, when we are saying about ovalbumin, a glycoprotein from the egg white, which will produce a peptide which is having blood regulation property. And talking about the classification of bioactive peptides, they are broadly classified into two, endogenous and exogenous. Endogenous are produced within our body, whereas exogenous can be obtained from the external sources. And talking about some of the external sources from which we can get the bioactive peptides, they can be obtained from plants, from the crops, cereals, and so on, and from the animals, especially venom of the reptiles, and from various food sources from egg, meat, and so on, and from marine organisms such as fish, algae. And why bioactive peptides are so important? Because it has multiple health benefits. It can act as anti-inflammatory, antioxidant, immunomodulatory, hypertensive, anti-cancer, and many more. So because of these multiple properties, they can act as an excellent scaffold for designing the peptide drugs. And as I mentioned before, the bioactive peptides can be derived from the parent protein. And one such protein our lab is constantly interested and investigating is hemoglobin. So everyone will be knowing about hemoglobin, which is the primary protein involved in the red blood cells. And looking into the structure of the hemoglobin, it consists of 4 subunits like 2 alpha and 2 beta chains. And apart from functioning as in their oxygen transport, they are a rich reservoir of bioactive peptides. So let us see how the bioactive peptides are derived from the hemoglobin. So once the hemoglobin undergo denaturations, it will become alpha globin and beta globin, and by the proteasomal degradation, it will become longer peptides. And oligopeptides will act on these longer peptides, and it will become the shorter ones. And 2 important classes of bioactive peptides are derived from the hemoglobin. They are hemorphins and hemopresins. So let us see in detail about these 2 classes of peptides. Talking about hemorphins, hemorphins are known as non-classical opioid peptides, which means that they have the ability in pain modulation. They can be derived from any chain of the hemoglobin except the alpha chain. And the property of these peptides is that they are very short, like 4 to 10 amino acid in length, and they all share a tetrapeptide sequence such as 5PWT, which composed of tyrosine, proline, tryptophan, and threonine. These amino acids will be present in almost all the hemorphin sequences. And talking about the hemorphin variants, we found out that the hemorphins is conserved almost all species, whereas an interesting variation was seen in camel, in which the glutamine has been substituted with arginine. So we were very curious to know what this variation actually do in molecular level. And these are the different physiological roles of hemorphins. Hemorphins can act as in regulating the blood pressure, in the pain modulation, in diabetes, cancer, and in the memory recognition purpose. So our lab has explored the molecular level of interaction of hemorphins on ACE, mu opioid receptor, and IRA, insulin regulated aminopeptidase. But we haven't explored the molecular level of interaction of hemorphins on diabetes and other opioid receptors, and so on. So these are some of the publications made from our lab which determines the interaction of hemorphins, both the camel and the non-camel one, on various drug targets. So here I am mostly interested how the hemorphins are involved in diabetes and pain modulation. So based on the early research report, it has been stated that hemorphins have an inhibitory effect on DPP4. However, underlying molecular mechanism of the hemorphins is unclear. Similarly, early research has demonstrated the opioid roles of hemorphins, and our lab has also explored the inhibitory potential of hemorphins, both the camel and the non-camel one, on mu opioid receptor or more. And however, the interactions with other opioid receptors such as kappa and delta opioid receptor are unexplored. Now I will take you to the next important class of the bioactive peptides, which is derived from the hemoglobin, which are chemopresins. Chemopresins can be derived from the proteolytic degradation of alpha chain of the hemoglobin, and they are first recognized as the modulators of the cannabinoid receptors, which is involved in the pain modulation, memory, mood, and so on. Here also we found the sequence variation across the species. Here is the picture of RBD chemopresin. Here we can see that in the some species we are having leucine in their sequence, whereas in the some species it is having the phenylalanine. Here also we are interested to know what this actually, the sequence composition greatly influence their activity. We are curious to know what is really happening in the molecular level. Just as the hemopresins have multiple function, and we are mostly interested in blood pressure regulation, because we have literature has stated that hemopresin exhibits hypotensive effects, and it has shown that intravenous administration of hemopresin led to lower blood pressure in the rats. And here we were curious to know what the variants of the hemopresin are doing into the hypertension. So based on all these studies, the studies conducted from our lab and also from the literature, we have reached to a hypothesis that hemorphins and hemopresins modulate the key enzymes involved in diabetes, chronic pain, hypertension through various molecular mechanisms, and this can be also influenced by peptide sequence variation. So understanding these variations, specific interactions at the molecular level will help to design peptide-based therapies. Based on that, I have correlated 3 research objectives. The first objective was in the context of diabetes, in which how the hemorphin variants, both the camel and the non-camel one, interact the DPP4, an important enzyme involved in the diabetes. And second was in the context of the chronic pain, how these variants are interacting the receptors involved in the pain modulation, such as delta opioid receptor. And finally, I have also analyzed the molecular level of interaction of hemopressin variants in hypertension by doing their interactions with angiotensin converting enzyme. And coming to the materials and methods I have used for this analysis, I have incorporated an integrated approach of both computational and experimental one. The first step was molecular docking. Molecular docking is a process in which how it will predict the binding orientation of a peptide inside the active site of the protein, which involves several steps such as target preparation, ligand preparation, and docking analysis. And for the computational analysis, I have used the Schrödinger suite of tools, which is a very important tool used for the drug discovery purpose. And the second step was binding energy calculation. So once I have predicted the binding mode of peptide inside the active site of protein, we have to determine how strong and stable our interaction is. For that, then we have used binding energy calculation, which uses Pratt-MMG in the Schrödinger. And finally, the molecular dynamic simulation. So the protein and peptide are not static in real life. So we will mimic the physiological condition to see how these complexes are behaving in our physiological condition. So we ran the simulation for a particular amount of time and to see how strong and stable our interactions are. So here also we involve several steps such as system preparation, energy minimization, and finally we do the trajectory run analysis. So whenever possible, I have complemented my study with in vitro assays, experimental such as enzyme inhibition assays, and we will calculate the IC50 values on this. So coming to my first study, which deals with the diabetes and hemorphins. So talking about diabetes, in a simpler term, we can see that diabetes is the increase in the blood glucose level in our body. And talking the statistics, we know that more than 500 million people are affecting the diabetes, which is expected to raise to more than 600 million in 2030. And most prevalent type of diabetes is type 2 diabetes. And here you can see the peptides I have used for the analysis. I have used 18 peptides, both the camel and the non-camel one, for the study. The protein I used for the study is DPP4. So let us see what is the role of DPP4 in regulating the glucose homeostasis. So DPP4 is also known as dipeptidyl peptidase 4, and its important role is in the regulation of incretin hormones. So once we intake the food, the small intestine will release the incretin hormones such as GLP-1 and GIP. The duty of this incretins is to stimulate the insulin production. So once the DPP4 is present, it will rapidly degrade the incretins. In the normal case, it is perfectly fine, but in the diabetic condition, we need to increase the stimulation of the insulin secretion. So for that thing, we have to inhibit this DPP4 molecule or protein. So DPP4 inhibitors allow the incretins to stay longer, and it helps in increasing the stimulation of the insulin secretion. And coming to the structure of the DPP4, the extracellular domain of the DPP4 consists of alpha, beta, hydrolase domain and 8-bladed beta propeller domain. The interface of these domains represents the active site of the protein. The active site of a protein is the region where the substrate molecule binds. And here you can see the important residues which are found in the active site. And these residues plays an important role in inhibitor binding. Some are essential for anchoring the peptide, some are involved in stabilizing the binding, substrate recognition and binding orientation and so on. So I have done the molecular docking of 18 peptides, and based on the glide score, binding energy and interaction profile, I find out that hemorphins H7, H6, H5 and their camel variants are having good scores, and out of this H7 is having better binding energy. So let us see in detail how these hemorphins are binding inside the active site of the DPP4. So here you can see the green colored one of ligand, the cyan one is the active site residues. This is the interaction profile of DPP4 and hemorphins, and we find out that in all the hemorphins we find out conserved core interactions in which the hemorphins are binding in a similar way inside the active site of protein. And this is the interaction profile of DPP4 and camel hemorphins in which we observe similar kind of interaction pattern, and also we have compared our results with a positive control, which is a diprotein. Diprotein is a potent inhibitor of DPP4. So we find out that the binding orientation also closely resembles the peptide inhibitor. And we have also analyzed the stability of this complex over a period of 500 nanoseconds using molecular dynamic simulation. And this is the picture of RMSD graph. RMSD stands for root mean square fluctuations, which determines the conformational changes happening to the backbone of the protein when peptide binds to it. And we ran in 3 runs to ensure the reproducibility and reliability of our results. And we found out that after the initial fluctuation, all the systems were found to be stable, which means that the peptide binding doesn't cause any conformational change to the backbone of the protein. And we have also analyzed the RMSF. The RMSF stands for root mean square fluctuation, in which residue of our protein is fluctuating more after binding with the peptides. And we find out that the peak regions represents the fluctuation, and we find the fluctuations are mostly seen in the loop regions. That is expected because they doesn't have the secondary structural elements, and rest all residues found to be stable. And we also analyzed the interaction profile of our protein and the peptide during a course of 500 nanoseconds, and we analyzed the polar and hydrophobic interaction for all the peptides. For the representative, I am showing only the top one, which is H7. And the key residues, which is the critical residues which is present in the active site of the protein, and our peptide is forming that interactions, are found to be stable throughout the simulation. And interactions were maintained throughout the 500 nanosecond simulations, supporting the fact that H7's potential as a potent DPP4 inhibitor. And coming to the key findings of the study, I find out that the hemorphins H7, H6, H5 and their camel hemorphins were showing high binding affinity to the DPP4, and out of this H7 is having higher interaction profile combined with the previous studies. And also they found stably bound over a period of 500 nanoseconds, and the core sequence YPWT contributes to the strong DPP-4 inhibition, and the binding orientation also resembles the diprotein A, which is a potent DPP-4 inhibitor. In summary, I can say that hemorphin demonstrated strong and stable binding to the DPP-4, providing first molecular level insight into their potential as natural inhibitors. And I'm happy to say that we have published this result in International Journal of Molecular Sciences. So coming to my second study, how the hemorphin variants are involved in chronic pain. So as we all know, chronic pain is a situation that is affecting millions of people. So morphine, codeine, phenyl are the frontline drugs which is used for alleviating the pain. So what really happens is that when we intake this drug, these drugs will bind to the specific receptors in our body. These receptors will help to reduce the pain. These receptors are known as opioid receptors. Mainly, three kinds of opioid receptors are seen in our body, which is mu opioid receptor, kappa opioid receptor, and delta opioid receptor, also known as MOR, DOR, and COR. So, talking about MOR is the primary drug target used by the clinical opioids. And apart from having a strong pain relieving capability, this receptor has a disadvantage is that it can cause a kind of addiction or kind of a high when we are using this drug target. And coming to kappa is mainly involved in depression, anxiety, and so on. Here, my focus was on delta opioid receptors. The delta opioid receptor is also having a strong pain relieving capacity, but when we are combining it with MOR, it doesn't cause a kind of addiction, which means that this drug target can be used to develop promising safe painkillers. So let us see the structure of the DOR. The DOR is a GPCR, which is a G protein coupled receptor, and these receptors will transfer the signals from the external to internal of the cell. And one of the striking factors of the GPCRs is that 7 transmembrane helices, and you can see the 7 transmembrane helices over there from TM1 to TM7. And this architecture will form kind of a bundle in which our drug molecule can bind. So the yellow one is the drug molecule. So it can exist in active and inactive state. In the active state, only the agonist molecule, the molecule which can enhance the activity of the receptor will bind. And in the inactive state, only the antagonist molecule which can inhibit the activity will bind. And I have done the molecular docking analysis using the 18 peptides which I mentioned before, and we have screened based on the glide scores, binding energy, and interaction profile. We find out that the camel hemorphins, especially 7 and 6, are having good scores compared to the non-camel one. So let us see in detail what is the real molecular interaction happening inside. So looking into the structure of the molecular interaction, we can see that docking revealed a similar kind of binding pose for all the hemorphins. Our peptides are represented in green color, the orange one is the active site residues. So here we can see it is a large hydrophobic pocket. The tyrosine 1, the end terminal of the hemorphins, is going deeper into the pocket, and it is forming a solid bridge and hydrogen bond interaction with aspartate 128. Aspartate 128 of DOR is very important. It is a conserved residue found all in the opioid receptors, and binding with these residues will help to activate essential for the activation of the DOR. And apart from that, there are several loops are there for the structure, and extracellular loop 3 also plays an important role in peptide agonist selectivity. So during the active state, the ACL3 will undergo a conformational change which exposes arginine 291. And arginine 291 is a specific residue which is found only in the DOR. And we find out that our hemorphins are interacting with arginine 291, which means that our hemorphins can specifically go and bind with the DOR. And looking into the molecular interaction of camel hemorphins, here we find out that, as I mentioned before, camel have a unique Q2R substitution which involves a positive charge in their sequence, and we find out that it is forming an electrostatic interaction with the aspartate 210. So this electrostatic interaction may have enhanced their binding energy or interaction profile. That's why maybe the camel hemorphins are working better than the non-camel ones. Here also we ran the MD simulation for 250 nanoseconds, and we couldn't find any major conformational changes. All the systems were found to be stable in all different runs. And coming to the RMSF, which determines the residue fluctuations. And as I mentioned before, most of the fluctuations are seen in the loop regions. Here, the most of the fluctuations are observed in the range between 239 and 261, which is the part of intracellular loop, and that is expected. And we have also analyzed the interaction profile of all the peptides, and this is the polar and hydrophobic interactions of camel H7. And we find out that all critical interaction, especially on the aspartate 128, all the things were maintained throughout the simulation, which means that our peptide is strongly bound inside the active site of protein. We had numerous hydrophobic interactions which helped to stably bound the peptide inside the active site of protein. And coming to the key findings of this research, I have found that the camel hemorphins H7 and H6 showed higher binding affinity than the non-camel ones. And we found a salt-bridge interaction with aspartate 128, which is a key anchor for the agonist binding. And N-terminal triad is mainly engaged in forming the hydrophobic and polar interactions. And the camel hemorphins are benefiting from Q to R substitution, where they form the salt bridge. And finally, this study offers molecular level insights into the binding mechanism of camel hemorphins with the dot. And we have published this result in Frontiers in Molecular Biosciences. And coming to my last and final study, which determines the role of chemopresence in antihypertension. So talking about hypertension or blood pressure, it is a global epidemic, and more than 1.2 billion adults are living with this condition, and it is the most common factor for the cardiovascular disease. And for this analysis, these are the peptides I have used. I have used both the variants of the chemopresence, both having the leucine in their sequence and the phenylalanine in their sequence. And here I have used, the protein I have used for this analysis is angiotensin converting enzyme 1 or ACE1. So let us see what is the role of this enzyme in modulating our blood pressure. So during the low pressure condition, when the blood pressure is low, the liver will produce the angiotensinogen, and to which the renin will act, which is an enzyme produced from the kidney, and it will change to angiotensin 1. Angiotensin 1 to the angiotensin 1, the ACE will act and it will convert into angiotensin 2. So angiotensin 2 is a peptide which causes the blood pressure. So coming to that, here another pathway is happening which causes the vasodilation. So balance of these two pathways are maintaining a normal blood pressure in our body. But in the hypertension condition, ACE1 is a drug target, the most inhibitors are designed. So inhibiting this ACE1 will help to reduce our blood pressure. So talking about ACE1, it is a chloride dependent metalloenzyme which has zinc ion in their active site, and it catalyzes the conversion of angiotensin 1 into angiotensin 2. And I will take you to the structure of ACE1. And two isoforms of ACE are seen in our body, somatic ACE and testis ACE. And this somatic ACE consists of an intracellular domain, transmembrane, and two extracellular domains, C domain and N domain. And C domain is mainly involved in blood pressure. So this is the active site of the C domain, in which the clinically approved drug is bound to it. The green color one is the drug molecule. And you can see the residue. These are all the important residues found in the active site, and you can see the zinc coordination as well. And for this analysis, first we have done the enzyme inhibition assays, and I have selected 8 peptides, and based on the enzyme inhibition results, we have shortlisted to 6 peptides. And from this dose response curve, we have calculated the IC50 value. IC50 value is the concentration of the peptide required to inhibit the activity of enzyme by 50%. And we find out that the lower the IC50 value, which means that potent the inhibitor. And based on this value, we find out that RADHPL, which is having leucine in their sequence, is having lowest IC50 value. And when we are looking into this result, we find out that the sequences, which are the peptides which is having leucine in their sequence, having lower IC50 value combined with the phenylalanine one. And we have also performed the molecular docking analysis of these 6 peptides, and our computational results has correlated with the experimental value, stating that RBD HPL is having higher glide score, binding energy and interaction profile. And let us see in detail how these are interacting inside the active site of the protein. So based on, it seems to be a bit crowded because the peptides are a bit long. So based on the interaction profile, we find out that the peptides which is having leucine in their sequence are interacting with the zinc ion. Also they are having extra hydrogen bonding, hydrophobic interaction and salt-bridge interactions. And we have also performed the molecular dynamic simulation for about 500 nanoseconds. As you can see, all the systems were found to be stable without major fluctuations. And coming to the RMSF, most of the fluctuations are seen in the loop regions. And we have also analyzed the interaction profile. We found strong hydrogen bond polar contacts as well as the hydrophobic contacts which that can be retaining throughout the simulations. All these factors implies that our peptide can bind strongly and stably inside the active site of ACE1. And we have also combined our binding mode of the peptides with the clinically approved drugs. The green one is our peptide and the orange and the pink one is the drugs which is approved by the FDA or clinically using ones. And the binding mode closely resembles with the peptide, and our peptide, since it is long, they can form extra interactions which also aid them to bind strongly inside the active site of protein. So key findings of my study is as follows. The HPL peptides are having favorable docking and binding energy, and they are having the lowest IC50 value as well, RBD-HPL. And why the leucine substitution is having better than the phenylalanine one? Because the phenylalanine consists of a bulky aromatic side chain which may have caused their fitting inside the ACE1 catalytic site, and also they may have reduced their steric hindrance. And also we find out that HPL variants are interacting with zinc ion and PACE1 residues. So HPL may be the clinically used inhibitors in their binding mode as well. And finally, in summary, I can say that they are promising natural candidates for the hypertension management. And we have also published these results in scientific reports. And talking about the limitations and the future studies of my research. So for first year analysis, we have done the computational prediction. To further confirm their activity, we have to do the in vitro or in vivo analysis. Will help to strengthen our work. And also we can optimize these peptides for the better stability and bioavailability. And talking about chemopresence and ACE inhibition, the third study, we have conducted the in vitro assays, but doing cell-based assays and also animal-based studies will help to strengthen our findings. And we have only considered ACE1 of the RAS. We are having other targets as well. So extending this study apart from the ACE1 will also helps to ensure the hypertensive activity of the peptides. And here also we can explore what structural modifications can be done in these peptides for their better stability and bioavailability. So I was fortunate enough to publish, apart from my PhD study, I was fortunate enough to publish 13 other publications, and I have some are from our lab and some are from the collaborations with other departments and colleges, and they are fortunate enough to publish all these things in the human journals as well. So before I conclude my presentation, I would like to say thanks to some of the people who stood with me during this journey. So first and foremost, I would like to thank God Almighty for providing me strength, resilience or patience to complete this journey successful. And I would like to thank my supervisor, Professor Ranjit Vijayan, for his unwavering support and guidance throughout this journey. As he mentioned earlier, I joined him back in 2014 as an RA. From there till now, he is the one who has helped me to shape me as a researcher. Thank you, Doctor, from the bottom of my heart. And I would like to thank my co-supervisor, Dr. Yusra, and her students, Aisha and Shama, for their endless support for doing all the experimental analysis. Thank you, Dr. Yusra. And I would like to thank the examiners, Dr. Vanajan from University of Malaya, and Dr. Sajid and Dr. Alam for your time and the feedback. And also, I would like to thank the computational biology lab members, Vinci, Amy, Abdul Rahman, for their enormous support during my journey. And also I would like to extend my thanks to all the faculty members of Department of Biology, College of Science, College of Graduate Studies, Dr. Ali Al-Masuki and his team for providing me fellowship during my studies, and UAE University for providing me funds and resources and for successfully completing this journey. And also I would like to thank some of the people who stood as a backbone during my journey. First and foremost, I would like to thank my parents. They are here. They came from India all the way to attend the presentation. So their prayers, their endless support, their motivation, all matters a lot. And I would like to extend my heartfelt gratitude to my family. I have two sisters and one brother. My brother is here. Thank you for being there for me, for encouraging me and supporting me. And I would like to thank my in-laws. They are not here. I hope they will be attending from online. Thank you. And I owe a special thank you to my husband, Mr. Jinari John. And thank you so much. And my kids, Johanna and Ryan, for their unconditional love. I love you both so much. And I also want to extend my heartfelt gratitude to my friends. First, I would like to thank Binsi. We were working together since 2012. Apart from a colleague, she's like a sister to me. She was there for me in all ups and downs. And also I would like to thank all the doctors of college. We were together during all my PhD studies. So even though they are from other department, they are here to support me. Okay, thank you so much, and all the other friends who are here. And last but not the least, I would like to thank Bhavani Chiji, who has taken care of my kids as her own. When I joined for the PhD, I was pregnant, 9 months pregnant. So from there till now, she's the one who has taken care of my kids. So I'm expressing heartfelt gratitude to each and every one of you. Thank you so much.
Priya, that was a very nice presentation, and I hope you have convinced the audience that you've done an exceptional job. Now the floor is open for a few questions. Do you have any? Does anybody have any questions? I'm sure you have something very that you might think is very trivial, but probably is not. No questions? Alright, in that case, don't go away. I'm told that there is no such thing as a free lunch, but Priya has arranged a packed lunch for us, so please stay outside for some time while the examiners ask her a few questions. And once we have come up with a decision, we will invite you back to let you know what the final verdict is. Thank you all for your time. Hope to see you all in very shortly. You think so? Very confident. I think that's one of the things the examiner has also observed. Very confident about how people present and work. So I think I have to say that the examiners were very impressed by the presentation, the work, and the way she answered the questions. But of course, nothing is perfect. There is room for some more improvement. So the examiners have given some small minor changes that need to be made before we can accept the thesis. So the examiners have said that they will accept it with minor revisions, and these must be made within 5 business days, as the university says. Yeah. So you have to complete the thesis with the changes that the examiners have said in 5 days, and then we will be happy to recommend that Mrs. Priya Anthony be awarded the degree of Doctor of Philosophy. Alright. So that is continue to submitting your updated thesis. So congratulations. Do you want to make any comments?
Congratulations for the wonderful work and your efforts and very impressive outcome. Good number of publications, although many of them are not from your PhD, but still they are relevant to your overall research. So I believe you have developed yourself, you have refined yourself, and you have knowledge about I am sure you will make progress into this, and you will find yourself working in such an area in a very good place. Wish all the best and good luck.
Thank you, Dr. Thank you so much.
As I said, I was very much impressed by your presentation. because you are like very new and in the department we are actually looking to do this, so it was confused, but overall it was very nice. I was very much impressed. You handling of the questions also.
Thank you all. And that's the formal end event. So Priya has completed most of the requirements to be awarded a PhD. And so thank you, Professor Manajan. We'll certainly be in touch. I'm sure we'll have an opportunity to talk and something. Thank you. You can thank
Thank you.